All right, so our next speaker is Anna Tyler, who's a research scientist at the Jackson Laboratory in Greg Carter's lab. And she'll be continuing this talk, but switching it a little bit more to epigenetics.
Sorry, could not figure out how to unmute myself.
Yeah, we can hear you fine. Yeah.
Okay. Okay. I got all this stuff away. Okay, yes, yeah, I'm Anna Tyler. I'm a research scientist in Greg Carter's lab at the Jackson Laboratory. I really appreciate being included, even though I couldn't make it there in person. Okay, let's go. Okay, so the project that I'm talking about today stems from the CUBE initiative at the Jackson Laboratory. And the name CUBE is derived from the idea of a multi-dimensional data space. So for example, here we have a genotype in one dimension, phenotype in another dimension, and environment in a third dimension. And the idea is in mice, where we have a lot of experimental control, we can measure along all these dimensions very densely, whereas in humans, our measurements are much sparser. So in the CUBE, the goal is to measure in the mouse data space densely, to learn patterns in the data, and then to use those patterns to learn, to predict patterns in humans, and then hopefully fill in the gaps of our knowledge in human disease. So the CUBE initiative was this large project that included 12 faculty labs across multiple JAX campuses, 11 shared scientific services, and of course, mouse services. There were 18 studies as part of the CUBE that all focused on type 2 diabetes. And we looked at inbred mice, outbred mice, CC recombinant inbred strains. We looked at multiple diets, multiple strains, both sexes, multiple tissues, and multiple interventions. And of course, we collected a lot of data. So the data are available in-house right now, but they will be curated and start to become publicly available as we start publishing. Okay, so the arm that I am going to be focusing on today is the diversity outbred arm. And this included 500 DO mice that were housed in Alan Addy's lab at the University of Wisconsin-Madison. They were both male and female mice. They were all maintained on a high-fat, high-sugar diet, and they were genotyped using the Megamuga array. We also have extensive clinical phenotyping on these mice. So we have many body weights, we have fasting plasma levels of glucose, insulin, and triglycerides, multiple oral glucose tolerance tests. And then at the end of the experiment, we harvested tissues and did bulk RNA sequencing on 4 tissues, so pancreas, adipose tissue, liver, and skeletal muscle. Also, islets were picked by hand for counting and measuring insulin content. Okay, so what do we want to do with these data? Basically, we want to put everything together into one big model that looks like this, where we have the genome as our causal element. The phenome is the clinically relevant outcome measure that we want to describe. And then the transcriptome sits between them, mediating the effects of the genome on the phenome. And because the transcriptome is gene-based and genes are annotated, it gives us a way to mechanistically describe the effects of the genome on the phenome. So for example, we want to say something like, there are variants in the genome that cause increased expression of immune genes in the adipose tissue. This causes inflammation and insulin resistance in the mouse. So our approach to doing this is very high level, very systems oriented. We're using a type of high dimensional mediation analysis that explicitly models that mediation model that I just described, the genome acting through the transcriptome on the phenome. It's an extension of canonical correlation analysis that can handle more than 2 data sets. So in this case here, we have each one of these pieces, each one of these squares is an n by n matrix, a kernel matrix that where n is the number of individuals, and then it describes the relationships between the individuals. So a kinship matrix is a kernel matrix for the genome. We do the same thing for the transcriptome and the phenome. And because we're using kernel matrices, our assumptions are really different from QTL mapping. So in QTL mapping, we assume that there is one or a few large localized effects in the genome that are affecting the phenome. Here, we're assuming massive polygenicity that's distributed throughout the genome. So it's a slightly different approach. And this method that we're using maximizes a specified network of correlations. So for example, here we want to maximize this path from genome through transcriptome to phenome. And then we want to minimize the direct path from genome to phenome. We're using the package RGCCA. And in the next section, Matt Mahoney is going to talk more about the technical aspects of this method and how we're developing it. So what we're basically trying to do is to identify latent variables for each of these pieces where the latent transcriptome perfectly mediates the effect of the latent genome on the phenome. So what we're looking for is the heritable part of the transcriptome that is also related to the phenome. Okay, so here are the latent variables that we get out of this model. So you can see, and this is the correlation structure between the latent variables. So the genome and the transcriptome are very highly correlated. The transcriptome and the phenome are very highly correlated. But once we account for the transcriptome, the genome and the phenome now have no correlation. So we got the latent variables that we wanted. The transcriptome is perfectly mediating the effects of the genome on the phenome. We wanted to know how easy this is to do in random data because CCA has a tendency to overfit. So we did some permutations to test this. We permuted the individual labels on the transcriptome before kernelizing and refitting the model. And then we created a null distribution of the path coefficient. So the path coefficient is the partial correlation of the genome and the transcriptome times the partial correlation of the transcriptome and the phenome. So here's our observed path coefficient, this red line here, and the null distribution is way far away. So it's actually really hard to get this, this high of a path coefficient. And if we plot the two pieces of the path against each other, we can see why. So here in null data, it's actually really easy to get a very high genome to transcription correlation, transcriptome. It's also easy to get a reasonably high correlation between the transcriptome and the phenome. But to maximize both of those pieces together is actually impossible, basically, in null data. And so this task is nicely internally regularized. Okay, so what can we do with these latent variables? One thing we can do is look at how the original phenotypes are loaded onto the latent phenotype. So here we see that it's mostly loaded onto body weight and insulin resistance, followed by some insulin measurements and some glucose measurements. And so what this means is that mice with very high phenome scores have very high body weight and high insulin resistance. So these are the more diabetic mice. And mice that have low phenotype scores have low body weight and low insulin resistance. And so these are the less diabetic mice. We can play the same game with transcripts. So we can look at loadings onto individual transcripts, which links individual transcript abundance to phenotype score. This is the distribution of transcript loadings in adipose tissue. So we see we have some of these strong positive loadings over here. So this means for these ones, if you inherit high expression of these transcripts, you tend to also be more diabetic. And if we look at the enrichment of these transcripts up here, we see a lot of immune activation, leukocyte migration. So if you're a mouse that inherits a reactive immune system, you also tend to be in the adipose tissue, you tend to be more diabetic. On the other side, we have the transcripts with negative loadings. So if you inherit these in high abundance, you tend to also be less diabetic. And these ones are enriched for metabolism. So we have fatty acid metabolism, branched chain amino acid metabolism, lipid metabolism. So mice that inherit high levels of these transcripts tend to be less diabetic on average. Okay, so one thing we can do with these loadings is just to take the strong loadings from each of the tissues. and then look at just cluster them across tissues. So that's what we've done here. And we can look for patterns. So the first pattern that you can see is that they're pretty tissue specific. So we have an islet specific cluster here, a liver specific cluster here. We can also look at functional enrichment in each of these clusters. So this pink cluster up here is enriched for leptin and leptin receptor interactions, which we know is involved in obesity and insulin resistance. This purple module is interesting. This one is enriched for apelin signaling, which is really integral to glucose metabolism. So glucose in the blood stimulates the pancreas to produce insulin, which then signals other tissues like adipose tissue and skeletal muscle tissue to start producing apelin. And apelin is a signal for these tissues to start importing glucose. So this reduces glucose in the blood. It also feeds back directly to the pancreas to tell it to stop producing insulin. In our population, the more diabetic mice tend to have high apelin signaling in the islet, but low apelin signaling in the other tissues. And so this suggests that mice that inherit low expression of this apelin module are then poised to develop insulin resistance on this high-fat, high-sugar diet. So we know from experiments that injecting apelin into obese insulin-resistant mice prompts their tissues to start taking in glucose. So it reverses the insulin resistance. And so that helps support the idea that inheriting low expression of this module might be causative in setting up the mice for insulin resistance. Okay, so there are lots of cool things that we can do with these data that I don't have time to get into, but I did want to touch on one thing that's related to the massive polygenicity assumption that we're using. And so rather than targeting individual transcripts with high loadings or even modules, we can target the transcriptomic profile as a whole. And we can do this using the connectivity map developed by the Broad Institute. So this is a database of cell lines that were treated with thousands of compounds or human cell lines. And they measured the gene expression responses to all of these compounds. And there's this query tool that you can use. You put in your own transcriptomic profile and you get back drugs or compounds that will reverse that profile. So we have our transcriptomic profile that's associated with diabetes or metabolic disease in mice. And so we can put it into this profile and see what comes back. And interestingly, we get back a list of drugs that's enriched for type 2 diabetes treatments. So this very top drug, PPAR receptor agonist, or drug class, one example is rosiglitazone that's used as a type 2 diabetes treatment to improve insulin resistance. It used to be used alone or in conjunction with metformin to treat type 2 diabetes, but it's associated with heart attack risk, so that kind of curtails its widespread use. Another drug class on here is sulfonylureas, that are associated. They're used to treat neonatal diabetes in patients that have KCNJ11 mutations. KCNJ11 is also on this list a little bit further down. So this drug class has, interestingly, also been used in patients with type 2 diabetes. And it performs similarly to insulin treatment, but has a reduced risk of hypoglycemia and weight gain seen with insulin. Okay, so I think this return of these drugs highlights a couple important points. One is that we used a transcriptomic profile that we developed in mice, and we got back drugs that are known to work in humans to treat diabetes. And so this is a win for the mouse-human translation. And then also, because we can, because these are our therapies that reduce symptoms of type 2 diabetes, it supports the idea that these molecular processes that we're identifying are causal in the pathogenesis of diabetes, because if we reverse them, we also reverse the diabetes. And then hopefully this drug list has other testable hypotheses for mechanisms and drugs that might not be under consideration right now. Okay, so in conclusion, we're developing a method, this high-dimensional mediation analysis, to identify molecular processes involved in complex traits by assuming massive polygenicity. We can identify transcriptomic signatures that mediate the risk of metabolic disease in DO mice. They're tissue specific, they're heritable, they're human relevant, and they're targetable. So I'm really excited about this method and the use of the DO mice, and then perhaps, paired with experiments, seeing more of what we can get out of this. So I really want to thank everybody on the CUBE team at JAX, especially Matt Mahoney. It was his idea to do this high-dimensional mediation analysis in this context, and we've been working closely together to develop it. He is there in person, so I encourage anybody who's interested to talk to him about this. And if we have any time for questions, I can take questions now.
Thanks.